In this lesson, we'll finish up the chemical level of organization by taking a look at two very large structures known as nucleic acids. So they name these nucleic acids because they were originally found in the nucleus of the cell, right? Hence the name nucleic acid. We're going to see that these two nucleic acids are really important for storing and also processing information in the cell. We'll get into how that's done in future lessons. Today, I just want to introduce to you the two main nucleic acids, their structure, that type of thing. So there are two nucleic acids. The first one is known as deoxyribonucleic acid. You probably heard of this because we usually just call it DNA. Right, a lot of us know about it because we use DNA testing to look at where we came from, from our ancestors, that type of thing, because DNA determines our inherited characteristics. Number two is important, the second dot. We're going to see that DNA actually regulates protein synthesis. So when we look at the cell anatomy, we're actually going to study that in great detail, how the DNA directs protein synthesis. It also controls enzyme production. And that's kind of the same as the first one. Remember that enzymes are proteins, so that's kind of the same thing. And DNA controls all of our metabolism. If you remember, metabolism is all of our chemical reactions at one time. All right, so that's our DNA. Ribonucleic acid, which we normally call RNA or abbreviate RNA, you may know or have heard of. Most people don't really know too much about this. The main thing for RNA for us right now, guys, is this: it directs protein synthesis. We're going to see that ribonucleic acid is maybe not direct, but is involved with protein synthesis. We're going to see there's three different types of RNA. All three of them are involved with protein synthesis. Okay, so those are our two nucleic acids, DNA, RNA. Let's take a look at how they're built. So if you remember from our previous lessons, proteins were made up of building blocks or monomers known as amino acids. Well, our nucleic acids also have building blocks. We call them nucleotides. So right here, nucleotide. These are the building blocks or the monomers of both DNA and RNA. And if you want to put in parentheses, those would be the polymers, right? Those would be the polymers. All right, so we're going to look now at the building blocks of DNA and RNA. There are three things that make up a nucleotide. The first one is a 5 carbon sugar. The name of that sugar is ribose. If you notice over here, ribose. Now, in DNA, the sugar is missing an oxygen. See right here? Deoxy means without one oxygen. So it's a 5 carbon sugar without, it's missing one oxygen. You don't have to know this, but it's on carbon number 2 it's missing. Where on RNA, the 5 carbon sugar has the oxygen, right? But nonetheless, whether it's DNA or RNA, the 5 carbon sugar is ribose. Okay? So it's a 5 carbon sugar. I'll show it to you in a second. The second part of a nucleic acid is the phosphate group. If you remember when we studied the functional groups, a phosphate group is a PO4. So this is going to be on there. Then there's going to be something known as a nitrogenous base. So on each nucleotide, there will be a different nitrogenous base. The names of them are adenine, guanine, thymine, cytosine, and uracil. Now, the first two are in a group of chemicals known as purines, and then the remaining three thymine, cytosine, and uracil, these are pyrimidines. We'll come back to this slide. Let's take a look at what the nucleotide looks like. So we're going to look for the 5 carbon sugar. We're going to look for the PO4, or the phosphate group, and we're going to look for a nitrogenous base in that nucleotide. So here's a picture showing us one full nucleotide. Let's start with the 5-carbon sugar. How do I know this is a 5-carbon sugar? It's a ring. At each corner, if you remember this when we did carbohydrates, at each corner is a carbon. So here would be a carbon, so that would be carbon number 1, carbon number 2, carbon number 3, number 4, and number 5. This kind of always reminds me of a house, and then there's a chimney sticking up over here like that. It's kind of the way I always see it. All right, so we have 5-carbon sugar. Right, in this case, this is known as ribose. Now, attached to the 5th carbon up here, this yellow, this is a simplified version, right? There would be a PO4 in here. This would be the phosphate group. So this is how this slide is showing the phosphate group in a simplified version. In DNA, this would actually form part of the strand of DNA. We're going to see that DNA is a double-stranded structure. So this is actually forming the strand. Then coming off of the strand, kind of into the middle of DNA, is going to be the nitrogenous base. So you'll see here this nitrogenous base is actually a 5 ring, I'm sorry, this is a single ring structure, right? We won't even get into how many carbons and that type of thing, nitrogens, we don't care about that for this course. We're just going to say this is a single ring. We'll see in a minute we can have a double ring nitrogenous base also. Okay, so 5-carbon sugar, phosphate group, right? They form like the strand of DNA or RNA. We'll get into that in a moment. And then projecting off of the strand, kind of into the middle of the DNA structure, we're going to see is a nitrogenous base. So adenine and guanine, as I mentioned, are part of this chemical group known as purines. Thymine, cytosine, and uracil are part of a chemical group known as pyrimidines. I just want you to notice one other thing I added to your notes. Thymine we only find in DNA. In RNA, where thymine would be, it is replaced by uracil. Right, so we only have that in RNA. This is going to become important, not so much in this lesson. This becomes important when I show you and we study protein production. That's something that we're going to have to remember. Of course, I'll remind you, but just to kind of introduce it now, that thymine is only found in DNA. Uracil replaces thymine in RNA. It's kind of the way I look at it. Let's take a look. So just to kind of give you an idea, here's our purines, adenine and guanine. Notice they're double ring structures, right? They have a combination of carbon and nitrogens. You do not have to know these structures. This is way beyond our course. If you ever take a biochemistry course, this is where you would study it. But you can see the two structures are basically the same, right? One difference here is we have an amino group where on that same carbon we have a double bond oxygen, and then over here is an amino group where we have a hydrogen. So there's just some slight differences. But adenine and guanine is what I want you to know. They are the purines. The pyrimidines are the other three: cytosine, thymine, and uracil. And again, if you notice, thymine only in DNA, uracil only in RNA. Notice these are also one ring structures. So when I was a student, I used to always mess up, which one were the purines, which ones were the pyrimidines. It hit me one day. Watch this. Pyrimidine has a Y, cytosine has a Y, thymine has a Y, right? So those are both pyrimidines. And then if you just remember that uracil replaces thymine in RNA, right, you'll know that these two have an association with one another. So it's easier to remember that cytosine, thymine, and uracil are the pyrimidines. Okay, and again, you do not have to know these structures. They're all very similar, which is minor differences between the three. Okay. So let's take a look at now again DNA and RNA, how they're kind of put together. DNA is actually two strands, right? Two pair of nucleotide chains here, or even if you want to put down strands, sometimes easier to remember. These are called complementary strands. So they complement one another. They're in a specific position next to one another, which I'll talk about in a minute. What holds the two strands together, if you want to give a little highlight right here, are our good old friends, the hydrogen bonds. So let's say you have a strand on the left, a strand on the right. They are held together by hydrogen bonds. We're going to see in subsequent lessons there are times, guys, where we're going to have to separate the strands. So those hydrogen bonds are going to be broken, the strands are going to be separated, work will be done, let's say for protein synthesis, and then once that work is done, guess what? The two strands come back together with new hydrogen bonds. The other thing you're going to see is the DNA, the strands, when they come down, they actually twist and they form what we call a characteristic double helix. So if you take a look over here, let's just say this is one strand this way here. We follow this one down like that. Here's the complementary strand like that. That's the second strand, right? And then you see these little blue lines in between right here. Those are the hydrogen bonds holding the strand on the left to the strand on the right. But notice it's forming a helix. You'll see there's like a big groove here. Again, this is a little bit advanced, but there's a big groove here and then a smaller groove here. That's characteristic. That's a classic look of DNA. And small groove here, major groove here. We actually call it major groove, minor groove. Again, you don't have to know that for here, but you can just kind of notice that as part of the twisting. Now, for our purposes, we're going to think of RNA as just a single chain. It gets a little bit more involved than that, but we're just going to consider it for our course as a single chain of nucleotides. And I just want to introduce to you today the three different types of RNA. In subsequent lessons, when we do protein synthesis, I will explain to you how they, or what they do, how they work. The first one I just want to introduce to you is known as messenger RNA. We usually abbreviate that mRNA. The second one is going to be called transfer RNA. We abbreviate that tRNA. And the third one is known as ribosomal RNA. Right? These actually help to form part of our ribosomes. We'll see in our next set of lessons, the ribosomes are a part of the cell where proteins are actually constructed. So just for today, again, anytime you hear RNA, think of protein production. We're going to see all three of these, right? One, two, and three are all going to be involved with the making of proteins. Now, we had said before that the DNA is actually two strands, but we call them complementary strands. That's because the nitrogenous bases, let's say on the left strand, are going to pair up with a specific nitrogenous base on the right strand. It's not random at all. And you're going to see this, guys. You're going to see, I always say A to T, C to G. Adenine is always going to base pair, I mean it's going to be opposite thymine. Cytosine is always going to base pair against guanine. So notice you have a purine base pairing with a pyrimidine, right? A purine base pairing with a pyrimidine. Right? So just kind of give you that type of thing. It's always going to be A to T, C to G, and I'll show you what I mean in a moment. And don't forget, on RNA there is no thymine. These other nucleotides will be there on RNA, except thymine will not be there. Uracil will replace the thymine. So what do I mean by that? Let's take a look. So over here, just to kind of give you an idea, let's say we'll take down over here. Say here's this strand on the left, and then here's the strand on the right down over here, and then over here we're going to blow this up so that you can see here's the strand going up and down on the left, here's the other strand on the right. Again, how do I know that? There's a 5-carbon sugar, right? Attached to that 5th carbon is the phosphate group. Remember we said that forms part of the strand, right? And this is just one building block here. This is one nucleotide. So notice, guys, off of this 5-carbon sugar here, this is actually deoxyribose, there's a cytosine. On the other strand, right, we have our sugar and our phosphate group, but here is a guanine. So notice cytosine base pairs with guanine. So now the left strand and the right strand are held together. See these little blue lines here? There's three of them. This is a, or these are, I should say, hydrogen bonds. Okay. Again, remember we said hydrogen bonds are weak bonds that hold things together, right? So we're holding this strand here with this strand, right? So if you notice, C to G. The nucleotides above it, right? Here's a nucleotide on the right, nucleotide on the left. A to T, A to T. So if you just kind of go up, you'll notice C to G, A to T, C to G, right? C to G again, A and T. Same thing, right? Normally it won't be different. It'll always be that type of base pairing. Up here, they're just showing you the same thing, the nucleotide on the left here, base pairing with the nucleotide on the right. Notice between A and T, you only have two hydrogen bonds. Between C and G, you have three hydrogen bonds. Just a minor structural difference. Okay, so that's this what we call the base pairing of the complementary strands. Now notice RNA, here's an RNA molecule. This only has, we're going to, for our purposes, say it's a single strand only. So you'll see adenine, guanine. It's not base pairing with anything because it's not a double strand. But again, notice what's missing. No thymine, right? What do we have in place? Uracil. Cytosine, guanine, adenine, and here's another uracil. So wherever there would be a thymine normally, there's just going to be a uracil in its place. Right? This is a great picture. They do a good job of that. So now that we know a little bit about DNA and RNA, I love this table here. This is a nice comparison of RNA to DNA. Let's take a look. Let's take a look at the sugar first. So we said that the sugar is a 5-carbon sugar. The name of the sugar in RNA is ribose. In DNA, it's also ribose, but we have to put deoxy because what is it missing? One, you got it, you probably said it, oxygen, okay? One oxygen. The nitrogenous bases in DNA, adenine, guanine, cytosine, and thymine. In RNA, same ones, adenine, guanine, and cytosine. So the first three are in both. But what's the one that's different? In RNA, we're missing the thymine and we're replacing a uracil. I love this next statistic here. Watch this. The number of nucleotides in a typical RNA, right? It can vary from 100 nucleotides stacked on one another to 50,000, right? So you can see these are large structures. But look at the DNA. Always more than 45 million nucleotides. That's how big these structures are. So the shape of the molecule for us, again, I'm telling you a little bit of a white lie, but we're going to keep it this way. We're going to just say that the RNA is a single strand. All right, it's a little bit more complex than that, but we're just going to say it's a single strand. But I do want you to know that the DNA is a double helix. All right, it's a double helix. And then again, as far as the function, RNA, or anytime you hear an RNA, think protein synthesis. It's always going to be protein synthesis where DNA again controls our genetic or stores our genetic information. It controls protein synthesis also, but it controls it where the RNAs perform protein synthesis. They're going to actually do the work. DNA is going to be more the controller. Okay, so that's what I want you to know about DNA and RNA. That'll give us a nice introduction, and then we'll build on them in future lessons. The last thing I want to talk about in chemistry is going to be high energy compounds, right? You may have heard of the fuel that we use in the body known as adenosine triphosphate, right? ATP. So why I bring it up here is that you're going to see that these high energy compounds like ATP, look at this guys, they are derived from nucleotides. So that building block of DNA and RNA is also part of our ATP structure. If I take a nucleotide and add more phosphates to it, that process would be known as phosphorylation. Right? So this is the process of adding phosphate groups to another molecule, right? And we end up producing this high energy bond. Let's take a look. I'm actually going to skip this slide for a minute. If you take a look up over here for a second, if I take our 5-carbon sugar, ribose, and I add a nitrogenous base, adenine, right? Would you agree those are part of a nucleotide, ribose and adenine? But watch this, guys. When we just have these two structures together, we call those two structures adenosine. Right? So they actually just changed the name. When they're combined, we call them adenosine. Now, if we add a phosphate, this would be adenosine. We add a phosphate, right? Phosphorylation. Now we would have adenosine monophosphate. Right? If you notice, this is a nucleotide, 5 carbon sugar, a nitrogenous base, and a phosphate group. So here's our nucleotide. Now watch this. If we add another phosphate, look at this bond here. They drew this a little bit different, more red, and they kind of made it a little bit wavy. That indicates this is a very high energy bond. This is actually a form of potential energy, that's stored energy. When we add this phosphate, watch this, guys, if we follow this bracket down, now we have adenosine diphosphate, right? This would be adenosine diphosphate, right? 1, 2. Then if we add another phosphate by phosphorylation, right, we would have adenosine triphosphate, another high energy bond. This is the one that we're going to be working with this semester. When this bond breaks right here, it releases a lot of energy for us to do work. Okay, so adenosine, when we talk about ATP, adenosine is an adenine and a ribose. We call that adenosine. Then we add a phosphate. We have adenosine monophosphate. That there is just a nucleotide, right? Three things make up a nucleotide. Now we phosphorylate again, and now we have adenosine diphosphate. This we usually abbreviate ADP. Then we add another phosphate on, adenosine triphosphate. Right, that's the high energy bond that we're going to be working with this semester. This is the same picture, guys, actually just showing it with the chemical structure. So you can see here's your ribose. Here's, and you can see on the second carbon, this is carbon number 1 right there. Here's carbon number 2. There's an oxygen. So that's how we know it's ribose. If that oxygen, excuse me, if that oxygen was missing, it would be deoxyribose, right? But this is actually with the oxygen. So that's how I know it's ribose. And then here's adenine, right? One of the purines. Right, and then we add our first phosphate to make our nucleotide. All of that would be the nucleotide. Then we phosphorylate a phosphate group. That would be ADP. Add another phosphate group. That would be ATP. Okay, we'll look at this again when we actually look at metabolism, how muscles, let's say, make ATP for muscle contraction. Okay, so this is just the verbiage of that picture. I like to look at the picture first, right? Adenosine monophosphate would be the nucleotide that contains one phosphate group. If we add another phosphate, we get adenosine diphosphate, or ADP. And then finally, adenosine triphosphate is ATP. This is what we're going to be working with mostly this semester. It's going to contain 3 phosphate groups. Now, this is again a little bit advanced. We're going to do this down the road. When we break this bond right here to release the energy, so we're going to convert ATP into ADP. We're going to actually break this phosphate off. The enzyme, where is it, right here, is known as adenosine triphosphatase. I usually just call it ATPase. This is the enzyme that actually causes the breakdown of ATP into ADP. Okay, so they do a good job with that. Good picture, kind of just showing the progression. All right, some more on that in future lessons. All right, so that concludes our actual discussion of nucleic acids and also our chemical level of organization. In the next few lessons, we'll actually now get into the next level of organization, the cellular level of organization.